So we're going to go ahead and get started. I'll give you an outline of kind of where we're going to head. I'll give you some intro info about dollar spot, kind of its background, its taxonomy, a little bit about kind of historical understandings about how we understand dollar spot and the various kind of traditional methods of control. And then we'll get into some of the current research that we've been doing on dollar spot and touch on some things there, particularly some molecular genetic work that Dylan's been doing. He'll share that with you. And then I'll kind of finish up with some comments about some products that are a little newer on the market for control of dollar spot and how they can be used and integrated into an overall management plan, and kind of wrap up there and then answer any questions along the way. Like Ed said, just pop them into that Q&A box and they'll go ahead and ask me the questions as we go. So we'll go ahead and get started. So if you're not familiar with where I'm at, and let's see, I can't. I'm going to see if I can. Did I get a pointer? Okay. All right. So I'll just leave it. I don't think you can see my pointer. At least I can't see it. So we're in the kind of the central part of this green ribbon here is the transition zone in the US. And so Knoxville's kind of in that little blip there between Kentucky and Tennessee. We're kind of right in the middle of that. And so we're right in the heart of the transition zone. And Dr. A.J. Powell liked to say that you can grow all the grasses equally poorly in the transition zone. And that's certainly something that we teach our students. You know, one of the challenges with being in the transition zone is that as you move a cool season grass farther south, you begin to push its ability to tolerate the temperatures and the heat of the summer. And as you move warm season grass further north, you start to challenge its ability to survive the winter. And so in the transition zone, every grass is kind of pushed towards the edge of its ability to adapt to environmental conditions. And as a result, at some point during the year, that grass is going to experience a bit of a challenge in terms of its ability to survive. And then that challenge also means that it's going to be at a weaker part of its physiology. And that's where it gets opened up to pathogen attack and things like that. So it's a great place for somebody like me who gets paid to kill grass on purpose to work. But it's a very challenging environment to maintain high quality turf grasses in. And so it makes for a great place to be able to study some of the challenges associated with our grasses. Now we're going to talk about dollar spot, but most of the time when I give a talk like this, I really like to start it out with kind of a little bit of a conceptual understanding of how I think about disease overall. And when I'm giving this talk in an audience or a room, I'll ask the audience what this is, and I'll often get somebody that wants to be the smart person in the room and say it's the disease triangle, and I'll make the joke that it's just a triangle. It only becomes the disease triangle when we add the labels to it, right? And so the concept behind the disease triangle is pretty basic. It basically says that if you have a suitable environment, a virulent pathogen, and a susceptible host together at the same place and time, that is when you have the potential for disease to develop. And there is a time component to this. It's important to remember that you can have you need to have somewhere in the ballpark of about 6 to 8 to 10 hours. It's variable, subject to environmental conditions, but somewhere in that ballpark, you need to have those conditions present in order for disease to develop. And the other thing that I want you to think about when it comes to the disease triangle is that we can think of the size of the triangle as kind of a conceptual potential for disease development. So if we don't do anything to modify the environment. We have a really susceptible host. And then usually there's not much we can do about the pathogen being present. It's present, it's virulent, so it's there. But if we have a really susceptible host, a really suitable environment, then that triangle is a pretty big area. And that means that the potential for disease development is pretty large and the severity could be pretty large. Now, if we were to plant a less susceptible host, then we would shrink the size of the side of that triangle down, and the triangle overall would become smaller. And the reason why this is a good way to kind of conceptually think about it is that most of our fungicides, those from FMC and others, are really good at controlling the diseases that they control. And generally speaking, all other things being equal, they're going to control somewhere in the neighborhood of 90 to 95% of the overall activity of a pathogen. So that leaves like 5% left over. Well, if you've not done anything to modify the host or modify the environment to shrink the size of that triangle down, then the 5% that's left over that the fungicide doesn't get, it could be a decent amount, and that could result in you looking at an area and going, gosh, this didn't seem to work. Whereas if you do the cultural practices, plant the more resistant hosts and things like that, then you shrink the size of that triangle down to where that 5% that's left over may be small enough that you don't really see any visible disease that you notice. And so it's a really good way to think about how all of these other practices that we encourage you to do in terms of managing disease are important to reduce the potential for disease development, right? So if this is just kind of showing like if we planted a resistant host, we'd shrink the size of that triangle down. And if we modified the environment, did something to the soil or improved our soil moisture management or soil fertility, then those might reduce the potential for disease as well. And that shrinks that triangle down smaller and smaller and gives our fungicides the best chance to actively or effectively control that disease. I've been looking a lot of sick turf this summer. This is just one example, right? And we've had a lot of warm days in Knoxville. I think we're at 20 out of 28 days so far since our first 90-degree day. The only saving grace is really that the low temperature has not come along with the higher temperatures. So we're still down in the low 70s for the most part. We've only had 2 days above 75 for a low. So that's a really good thing because it's often the low temperature that's the problem, not so much the high temperature. And so we've had a stressful summer so far, and it looks like it's going to kind of continue. So all of these cultural practices that we can engage in will help turf manage these kinds of conditions better than they would if they weren't done. And so I would encourage you, whether we're talking about dollar spot control or talking about other diseases or even just the stress of summer, that you want to engage in those cultural practices that are going to help manage that turf through a stressful period. Okay, so let's get into kind of dollar spot and dollar spot history. So, dollar spot has been a disease that we've known about for a long time. It was originally called little brown patch. It was first talked about by Monteith in 1927, and then formally characterized by F.T. Bennett in 1937, so about 10 years later. And one of the things that puzzled both these individuals was why dollar spot remained fairly small in size compared to other diseases. So at the time that Monteith called this little brown patch, there was also large brown patch, which is the brown patch on cool season grasses that we think of today, caused by Rhizoctonia. And Bennett in 1937 characterized the fungus and placed it into the genus Sclerotinia and called it Sclerotinia homeocarpa. And for the next, let's call it 55, 60, maybe even 70 years, we pretty much knew that it wasn't properly in the Sclerotinia genus, but we didn't know where to put it. There were various attempts to try to put it into one genera or another, but it was it wasn't very clear where it belonged. And so it made it a real challenge to manage. But in terms of the taxonomy, but the symptomology and some of the basic understanding of how this pathogen develops and grows, this has been pretty well understood for quite some time. And one of the things that's always been perplexing is why the diseased area stays so small. And we'll talk about that a little bit later on here. So then in the 90s, there were some genetic experiments done to show that it clearly didn't belong in Sclerotinia, but it might belong in something like Poculum or Malera discus or Lanzia. There were a bunch of different genera that are all somewhat related. They're all still very closely related to the Sclerotiniaceae in terms of the families, but it was just not really sure where it officially belonged. And then in 2012, there was some work by Liberty who showed that there were kind of two types that he identified, and they found this F type was only found on warm season turf, and this C type, is what he called it, was found on both warm and cool season. And there seemed to be these two different types, and that they had slightly different morphologies, but they looked very similar under the microscope and all of those kinds of things. And so it was the first bit of evidence that maybe there was something going on with warm season versus cool season, and so on and so forth. And then in 2018, we finally get kind of the seminal work done to show that conclusively where dollar spot belongs in terms of the taxonomy of the fungi. And so Salgado Salazar in 2018 with others reclassify this into its own genus called Clariredia. Redia, named after Reed Funk, the turfgrass breeder at Rutgers. And so they renamed the genus or named the genus Clariredia. And the only fungi that are in Clariredia at present are dollar spot fungi. They found multiple species of Clariredia. The four that they particularly laid out were Clariredia homeocarpa, which is found on red fescue grasses in the UK only. There's Clariredia benneti, which is found on cool season grasses in the UK, the Netherlands, and the US. And then the two most predominant species, Clariredia monteithiana and Clariredia jacksonii. Monteithiana, named after Monteith, the guy that described it initially in 1927, found on warm season grasses worldwide. And then Clariredia jacksonii is found on cool season grasses worldwide. And so since there's also been some work done to identify two other species, Clariredia paspali and Clariredia hainanins, that have been discovered in China. So basically, you've got kind of 6 species. All of those species are in the genus Clariredia. The only organisms in Clariredia are grass pathogens that cause damage on turf grasses and cause what we call dollar spot as a common name. So these species can infect all different types of turf grasses. Most can infect cool season grasses. The largest proportion of those is jacksonii. They will damage cool season turf. And then you've got Monteithiana on warm season turf. And one of the things that comes up quite frequently is there's a fair number of lookalikes in terms of dollar spot, from spider webs on grasses to cottonwood tree seeds. a lot of times people will look at something like this and go, oh, wow, look at the dollar spot really fuzzing up on the turf. And so one of the things that I want to point out to you here, and it's I do it by way of kind of making light of the situation, but when we look at an area that might have dollar spot and try to determine if it is in fact the fungus that we're looking at, what we're looking for from a pathology standpoint is we're looking for lesions or disturbed or damaged turf associated with whatever it is that we're seeing, like this fuzzy area in this image. And so we're going to look for classic lesions that are associated with that white fuzz, and that will help us confirm that it is in fact dollar spot. In this case, these are just cottonwood seeds sitting on top of the turf, but they look very much like the fungal mycelia that you're going to see in a second. And so it's important to kind of clarify and make sure that when you're looking at an area that you think might have dollar spot, that you see not only these threads and white fuzzy growth, but you also see lesions and damaged turf that kind of help confirm that that's in fact what's going on. So this is more like what you would see on a higher cut lawn. Dollar spot tends to occur over a larger area. That's largely because dollar spot tends to be pretty sensitive to ultraviolet light. I'll let Dylan touch on just for a moment when he talks about some of his molecular work, some of the other work that he's doing with ultraviolet light to better understand dollar spot and how it's affected by ultraviolet light. But in taller canopies, if you're in a lawn care situation, you're going to see larger areas of mycelial growth. And that's largely because of the shading and the microenvironment created by that higher canopy of turf. But the symptoms on the leaves are going to be very similar to what you see on a shorter canopy. But you definitely are going to see, like you see in this image, areas of white fuzzy growth, but you also see tissue that is not doing so well and is clearly displaying symptoms of attack. And as the disease progresses, you can see some thinning. You continue to see some fuzzing as the mycelium comes out and grows under favorable environmental conditions. But this is very typical of the symptoms that you would see on a higher cut canopy, like in a lawn care situation. If we go down to a shorter canopy, like a shorter fairway height, we tend to see a more distinct spot. And as we continue down to, say, something on greens height, that spot becomes more and more distinct. But you still can see the fuzzy mycelium under conditions of high humidity. But again, in this image, you can see behind that fuzzy white growth coming out of the tissue, you can see brown desiccating tissue that's clearly showing that it's being affected by whatever it is that is growing out of the turf right here. And then as we go down to a putting surface, the spots become very distinct, and you generally see a large number of localized spots that can coalesce into larger areas and cause damage to larger areas. And the big issue with dollar spot, of course, is that as that disease progresses, you will get a uneven surface that can result in bumpy ball roll and things like that that are not favorable for playing golf on those surfaces. So it's one of the more widely associated diseases around the world, and is also one that probably has more fungicide dollars spent on it in terms of control, because the period that the disease can be active is a pretty long period of the year, and you can see it on multiple heights of cut. So as a result of that, there's a lot of money that gets spent trying to control dollar spot, and it's an important disease on lots of different turf grasses. This is very kind of robust mycelial growth coming out of the turf on a humid morning. You can see it's white and fuzzy, and then as the day goes on, the turf will dry out. The white mycelium will kind of go away, and you'll just be left with a kind of a circular area of brown tissue that we call dollar spot. In those areas where we have a lot of disease activity, and that disease activity goes on for some time, you can see these areas start to lose some surface integrity, and they get more pitted and sunken. And then that results in even worse ball roll than maybe the superficial kind of phase of the disease where it's just in the leaf tissue. And as that turf gets pitted out, you'll see openings. Then that opens up the canopy for weeds to germinate. You'll see sometimes in really severe outbreaks that aren't managed, you'll see some goosegrass germinate in these areas and start to fill in. You can see crabgrass germinate and fill in, clover, all sorts of different weedy species that might germinate and grow into these open voids. So it not only creates a problem from a surface playability perspective, but it can also create a problem in terms of managing the turf stand and allowing other weedy grasses or plants to get in to the mix and start causing problems, which then leads to challenges with managing weeds in addition to diseases. But this pitting condition is very typical as disease progresses. The other thing that you see in this image that's kind of evident is that you'll see some clustering of spots in areas where the turf is very susceptible. We don't fully understand this. There's some hypotheses that maybe a particular clone of a synthetic variety of bent grass is more susceptible, and so that's why you see more spots. But it's not fully understood why we see these clusters of areas. Like you can see kind of in the upper left, a real tight cluster, and then there's not a whole lot of disease, and then you see little areas of more spots, and then in between less spots. And so we do see this clustering from time to time. And this is just a close-up showing kind of that pitting of the turf, where you not only do you get the foliar damage, but it will go all the way down towards the soil surface and degrade the integrity of that surface to a point where the ball roll becomes bumpy, and it becomes very challenging to manage that way. We can also see dollar spot on warm season turf. I mentioned that when we talked about the taxonomy, but we do see it on zoysia grass. So this is on zoysia grass. You can see the symptoms look very similar to brown patch symptoms on higher cut turf. You'll see a darkened margin with a light brown kind of band. Oftentimes, one of the distinguishing features for dollar spot on the higher cuts of grass is that band will kind of become sunken, and it'll look almost like an hourglass kind of straight across a leaf blade. So that can help discern between this and something like brown patch. But it can often be difficult to tell which one is which in certain situations. So it's important to make sure that you get a good diagnosis if you're not sure. But usually in a situation like this, where you can see plenty of white mycelium, you kind of see a small spot, that's usually enough to be pretty confident that you're dealing with something like dollar spot. This is dollar spot on Bermuda grass, and oftentimes on Bermuda grass, it's viewed more as a nuisance type disease. You'll see it in areas where fertility is lacking. We'll talk a little bit about that in a few minutes. But you'll see it in areas where fertility is lacking. It's not doing so hot. It's slowly growing. And then that opens up the opportunity for the pathogen to infect and cause some spotting. And you'll see those localized spots like you do on something like creeping bentgrass or Kentucky bluegrass. One of the other things that's happened in the last 40, 50, 60 years is we've had a really, significant move in the breeding world to breed varieties of bentgrass that are more resistant to dollar spot. This is just one example, but Dylan and I were over in Rollsville, North Carolina for the pure seed testing field day, and we had the opportunity to look at their variety trough for their bent grasses and saw anything from pen cross that you see here in this image. You can see how much coarser textured it is. It's more open in terms of its canopy. And just next to it is pen A4, which was kind of the next generation of bent grasses that came out after pen cross. And you can see how much more dense that turf is, how much more finely textured it is. And that results in a better putting surface. But then at the same time, we've also seen varieties developed with more resistance to dollar spot. And so that's a great thing. And so now the big term is super bents. And the super bents are those that have this fine texture combined with these characteristics like an increase in disease resistance. And so you're getting things like 777, Declaration, 007, L93XD, Oakley, Piper, the Pure series, Pure Distinction, and others that are both resistant to dollar spot as well as other things, and very fine textured that allows for a very tight mowing height and very high putting quality. And so that it's kind of it was very exciting to see this kind of up close and personal and just really see the progress that's been made over the last 80, 90 years to go from something as coarse and as rough as pen cross and get into this era where we have bent grasses that are extremely fine, but also pretty resistant to some of our more important diseases. So that's kind of exciting to see, and certainly something that gives us the potential as turfgrass managers. If you're in a situation where you're choosing to be able to make a choice about the genetics that you're going to employ on your putting surfaces, that you have that ability to do that. That's a pretty cool thing. So as the disease progresses, so I showed you some pictures kind of showing this, but it's important to understand that as the disease progresses, it typically occurs in high relative humidity environments with moderate temperature. And Ryu and others in 2021 did some work looking at the progress of dollar spot microscopically. And they basically showed that you get your first sign of disease as kind of the white mycelia appearing as like a cobweb on areas of turf. And if you look at those lesioned areas, what you find is that this fungal hyphae actually get into the plant by penetrating through the stomates of the leaf. They will form an infection peg and then push those cells apart and kind of make their way in. And in the process of that, they will secrete things like oxalic acid and other cell wall degrading enzymes to break down the cellular structures, the waxes and things that are designed to keep organisms like dollar spot out. You can kind of see that in the right image. In the left image, you can see it actually going through a stomate. And in the right image, you can see those infection pegs kind of pushing down to start getting into a plant cell. And so that's kind of how the fungus first starts to get into the plant. And then when you start to see symptoms, I mentioned that hourglass shaped lesion. This is typically what you'll see a straw colored area bounded by darkened borders that show where kind of the pathogen activity is. And at the end of the day, on shorter cut turf, you're going to see these silver dollar sized areas that have a brown color to them, and then if it continues, you'll see that area kind of get pitted and start to sink down, which then causes a challenge with the uniformity and playability of a putting green or on an athletic field. So one of the other questions that's been kind of postulated, in addition to why does it stay so small, is, well, how does it spread? Because this fungus and the several different species that belong in the genus Claridia they don't produce spores. They don't go through sexual reproduction very easily, and they don't produce any kind of asexual spores called conidia to spread from one place to another. So the question's always been, well, how does it get from one place to another? And various hypotheses have been, kind of bandied about over time. You've got the idea that you keep it on your shoes and you spread it around as you walk from location to location, and that's certainly probable or possible, but doesn't explain the widespread movement of the pathogen all over the place. And so the best hypothesis is that it's pretty clear, and we know that we do this when we establish a new study area. In fact, we'll allow some dollar spot to develop. We'll collect the infected clippings and we'll spread them around. And so that's basically spreading the mycelia, the body of the fungus, from one location to another. And that's pretty much how it spreads. And that's the best explanation that we have for how it spreads today. We've shown my research that I did for my PhD showed pretty clearly that the equipment, whether it's the mowers or hoses or whatever, don't do a very good job of moving the pathogen. They probably move it a little bit, but if you're collecting the clippings themselves in a vessel, you're probably not spreading the organism very far on your equipment. Whereas you might do that with other pathogens like Pythium or things like that. So one of the other questions was, well, when anybody has ever looked at doing any of these genetic methods to study dollar spot, one of the things that's been consistently found time and time again is that no matter where in the world we sample this organism from, we tend to see that it looks genetically very, very similar to each other. There's not very much difference from one individual to another. Whereas, you may be familiar with using DNA in like a court case to identify a perpetrator versus a non-perpetrator. It's not too hard, even within humans, to identify places where there's some differences and show that, this more likely than not, this person versus that person. Well, it was very difficult to do that with dollar spot. Everything looked like it was its own genetic clone. It was almost identical. And so Ryu in 2014 did some work and found that and one of the things that came up in these studies was that there was a discussion of something called a founder effect. And the founder effect basically means that the organism has to be coming from basically the same place, which is why it's all pretty much the same. And so one of the questions was, okay, well, where would this place be? Where would it be coming from? And so Ryu looked at commercial seed and found that in 8 of the 12 lots of seed that was sampled, they were able to find dollar spot in that seed lot. And so it wasn't very much. It was very low frequency, but it was present in the seed. And if it was present in the seed, then that would mean that if you sold that seed all over the world, that all of those individuals that are very similar are coming from basically one place, the seed fields, and they're going out into the world. And then you're seeing it develop over time in those areas. Dollar spot also overwinters pretty well, so it can survive the weather pretty well. As it gets colder, there's less survival, but with more snow cover, you tend to have more survival. So there's a little bit of a catch-22 there. One of the hypotheses for why iprodione resistant organisms for dollar spot tend to, if you move away from iprodione, you tend to be able to come back and use it in the future. Whereas some of the other resistances like DMI resistance and things like that, they tend to stay more present, is that iprodione resistant individuals tend to be able to not survive as cold a winter. And so a lot of times they struggle to survive conditions that might otherwise be okay for the organism. And as a result, they kind of disappear from the population, and then you can go back and use the mode of action. So this is this is kind of basically where we've ended up, is that we kind of think that this organism tends to move in infected seed, and then as a result it spreads all over the world via the sales of seed, and that's really one of the reasons why this ends up being kind of similar all over the place, is that it's coming from a similar location. And so this is just a graph that shows that presence of organisms, and the presence of the organism of dollar spot in it. We talked a little bit about how disease develops in terms of conditions. So certainly relative humidity needs to be high. So oftentimes when you've had a rainy period, you've got plenty of soil moisture, you're high humidity, you've got a lot of dew. Dew is full of nutrients, and so the fungi use that as a carbon source, and they're able to grow using the dew as a nutrient source. Moderately warm temperatures, so 21 to 27 degrees C, that's going to be in your mid 70s, low to mid 70s. So that's a pretty optimal period of time for the growth of the pathogen. It can be active across a very broad range of temperatures, so anything from 15 to 32. So 15 is like in the low 60s, and 32 is going to be up around 90. So pretty much the whole range of weather conditions under which we're growing cool season turf, this organism can be active and growing. We typically see it as flushes of disease in the spring, and then in the fall. Low soil moisture tends to increase the severity of the disease, as does low fertility. So it's certainly something that that low fertility can dramatically increase disease. In fact, it's one of the things that I do in my trials to try and encourage dollar spot to develop, is that I will actually make sure things stay relatively dry, and that we don't put down too much fertility in order to get disease. And this image kind of shows that. The blue paint line there separates our brown patch trial from our dollar spot trial, and you can see how much more prevalent the dollar spot is on the right of that blue line versus the left. It's not to say that it's not present on the left. You can certainly see some spots, but it's nowhere near as significant as it is on the right, which really kind of highlights how important fertility is in terms of the development of this disease. And then I mentioned early on about the chemical controls. So we talked about dew, removing dew, whether it's through rolling or dragging, doing some sort of dew whipping on fairways, things like that. All of those things are good cultural practices to reduce the potential for disease development. Certainly, the organism is able to overwinter kind of in the organic matter and thatch of a turf canopy. So sand top dressing, regular vertical mowing, and proper nitrogen fertility is going to help with the cultural practices to manage thatch development. And then that's going to help reduce or make an environment less conducive for a lot of inoculum to be present. We talk about how important that fertility level is, to make sure that it's moderate, so that it's enough to support the growth and keep growth kind of moving forward. And it's not so limiting that the plant is struggling to grow, which then just makes it more susceptible for disease development. And also really important to manage soil moisture. One of the things that I've noticed in the last probably 15, 18 years is how important it is to have that TDR and be familiar with where, what percentages at your particular facility constitute a an appropriate amount of moisture. And that's generally going to be when you filled about 50% of your pore space that is available to plants up with water. So you want about half of the pore space full of air and oxygen, and you want the other half with water that's available for plant growth. And so in most sand situations, that's going to be a soil moisture that's somewhere in the 16 to 22% range from a volumetric soil content, water content from a TDR. And if you're maintaining moistures that are in that moderate amount, that gives you the ability to have a plant that's not suffering drought stress and making itself more susceptible to dollar spot pressure. From a traditional standpoint, there's always been and continues to be a lot of options in terms of fungicides. The modes of action for most of our fungicides, even today, are in systemic fungicide classes like the SDHIs, the DMIs particularly, and then you have your dicarboxamides and contact fungicides are kind of the four main areas in all of those systemics, not the contacts, but all of those systemics have had resistant populations reported in the field. And so certainly managing, alternating, rotating your modes of action is really important to getting good quality disease control from these various fungicides. And then the other thing that's happened, and I was part of a group that helped kind of ground truth the development of this model, the Smith-Kearns dollar spot prediction model, is that we have a predictive model that does a pretty good job of helping you plan when you need to spray a fungicide. And it basically relies on a 5-day moving average of relative humidity and temperature. And it specifies the likelihood of an outbreak or an epidemic. You set a threshold. So most people you will use the 20% threshold, and you can adjust that up or down subject to your tolerance. If you're a low budget facility that can tolerate a little bit of dollar spot on a fairway, you might bump that threshold up a little bit. If you're at a place that's a very high end place that can't tolerate much dollar spot at all, without people losing their minds, you might bump that down a little bit. But you can set that threshold. And basically what happens is when the model goes over that threshold, it recommends you spray. And so if you've already made an application and you're within the window of control for that previous application, then even though the model is saying that there's the potential, you're protected because you've sprayed a fungicide. If we get to the end of the interval of the fungicide that you're applying and you find that the threshold is now below that, then you don't need to spray and you can delay until it gets back above, and that gives you a little bit of time. And if you add up those little bits and pieces of time, you might end up saving a spray or two over the course of the season. I'll go ahead and introduce Dylan McCallum. Dylan is my master student. He's been working on this project looking at dollar spot and bent grass and how dollar spot develops in bent grass over time. And we've been using a pretty sophisticated technique called RNA-seq, which is basically where we sequence all of the different little pieces of RNA that get put off as a biological process is happening to turn things on and off in the cell. And then we can harness that to understand what's being turned on, what's being turned off, and what their functions are, and help kind of hint at maybe there's some places where we can either get some more resistant grasses or we can identify new targets for controlling disease. So I'm going to let Dylan take it from here and kind of tell you a little bit about his research.
Thanks for that lovely introduction. Hi, everybody. My name's Dylan, and today I'll be talking to you a little bit more about what I am doing for my master's. So the question we're trying to answer specifically is we want to know, are there gene expression differences between creeping and colonial bentgrass cultivars when we infect with dollar spot? So colonial bentgrass isn't used as widely in the golf course industry due to its inability to spread rigorously. It's a bunch type grass. And so as a result, it's not selected for as great or as highly on golf courses. But what colonial bentgrass has that creeping doesn't is that colonial bentgrass is far more resistant to dollar spot in the field. And so these two are both in the same genus, and so they're very similar in the ways that their genetics work, but there may be some differences in gene expression that give us that different phenotype. So my hypothesis for this study was there will be up and down regulated genes over the infection period that are critical for pathogen identification and resistance in each bentgrass selection. The colonial bentgrass specifically will have defense genes that are expressed at a higher amount or even unique from the creeping bentgrasses. And so our objective was to evaluate the time course of the bentgrass dollar spot pathosystem. So looking at how does gene expression change over time during the infection for both the creeping and colonial cultivars. And like Dr. Horvath said, we used a technique called RNA sequencing. The other one is to evaluate the differential response by bentgrasses with a range of known resistance to dollar spot via challenge inoculations. So with that, we'll move into my study design. So with my study, we have two cultivars of creeping bentgrass. We have Declaration and Crenshaw. Declaration is one of the more resistant cultivars of creeping bentgrass to dollar spot, and Crenshaw is considered our susceptible population. And then we have one cultivar of colonial, which is BCD, which is considered one of the most resistant cultivars of a colonial and far more resistant than any creeping bentgrass cultivar on the market today. Additionally, we sampled at 4 different time points. So we sampled at 48 hours after inoculation all the way to 96. And in addition, we also had non-inoculated controls at the first and last time points so that we could compare our gene expression to what's been infected versus what's not. Essentially, our goal is to find genes that contribute to colonial bentgrass being more resistant to dollar spot than creeping. Specifically, we want to help breeders further along their goal of creating new cultivars of creeping bentgrass that are more resistant. And we were able to go down to Rollsville, just like Dr. Horvath said, and see how far the genetics has come with creating more finer textured turf. And so we know that we can do the same thing, but just make more dollar spot resistant cultivars. One of the main challenges with this kind of data set is just the breadth of it. So the total number of genes that I sequenced in my RNA-seq experiment was, and we averaged about 83,000 between the 3 different genotypes. And so what the challenge is we have to take all those genes and weed them down to get to the point where we only have maybe 10 to 20 that are actual gene targets that we can give to breeders in order for them to select new cultivars of creeping bentgrass and hopefully have higher resistance to dollar spot. So when we got the data back, we kind of split our analysis into 3 different pathways. So the first pathway, we wanted to look at, okay, are there any genes in the colonial bentgrass genome that are unique that give it a higher resistance to dollar spot than the creepers? The second pathway was looking at what genes they share because they're in the same genus. And so actually 2 of their genomes are shared. And so we can look at, is there a different expression between colonial and creeping? And so maybe there's some genes that are being upregulated at a higher rate in the colonial, and that's conferring resistance in the field. The last one is the last pathway was looking at genes that are always on or constitutively expressed in this shared population of genes. And so if we look at some of the results that we got, this is looking at just unique transcripts within the colonial bentgrass. And so looking at this donut plot here on the top left, you can see that 56.1% of the genes I sequenced were unique to the colonial bentgrass versus the other 44 or 43.9% being shared either with declaration or Crenshaw individually, or shared between the both creeping bentgrass cultivars. And so if we break down that 56.1% a little bit further, we can look at what pathways these genes are associated with and then filter out the ones that are more interesting to us. And so some of the interesting ones we got were obviously defense response, looking down here in the bottom right in the bar graph. But we also saw things like chitinases, nutrient reservoir and oxidative stress, and pattern binding. And all these pathways are all critical for defending against pathogens. And so now we can take the 20 or so genes in each one of those and look a little bit deeper and see if those genes are maybe conferring the resistance that we see in the field. Looking at the shared genes, I was able to determine and look at gene expression between the different genotypes with these shared genes and pick out ones that were expressed at a higher proportion in the colonial than the creepers. And so these genes maybe are just having a bigger effect inside the cell and allowing for that colonial bentgrass to fend off the fungus a little bit easier. And one of those is down here on the right, called callose synthase 8. So callose synthase, callose is essentially a compound that toughens up the cell walls of a plant cell and doesn't allow penetration by the fungus. And so if you remember from a couple slides ago, we showed that the fungus dollar spot uses infection pegs to kind of penetrate through the cell wall and get into the cell so that it can extract the nutrients. With callose, that's preventing those infection pegs from even getting through the cell wall and then thus keeping the plant cell happy and alive. The last pathway we looked at was constitutive expression, or genes that are always on, even when there's no pathogen present. And so what I was able to do is take those control plants and compare the expression levels between the different genotypes, so between the creepers and the colonial bentgrass, and using the same pathways and the same terms, I was able to pull out specific genes that were related to chitinases, defense response, oxidative stress, and pattern binding. And so down here on the right is kind of a blown up version. And so we go down from, what did I say, 83,000 to a couple hundred really quick this way. And now we can look at these more in depth and see, is there a different response, or are these the reason why we see more resistance in the field for colonial? And so the end goal of all this is essentially to come up with some gene targets that we can give to breeders and allow them to breed new cultivars quicker, because the current timeline for a new cultivar is 10 to 15 years. And so if we can get that down to 5 or 6 years, that allows us to have more access as superintendents or as architects to use those new cultivars that are more resistant and potentially help save a fungicide spray here or there during the year.
Because I'll wrap up just a couple of things with fungicides, just to kind of touch on some fungicides that are useful and that question about the dollar spot and brown patch. So and thanks to Dylan for that presentation on some of his work. It's been kind of exciting to see some of the stuff that we're learning from that. But Densicor, certainly a newer DMI. Excellent on dollar spot and brown patch. So that's a good example of a fungicide that's going to pick up both dollar spot and brown patch. Also has other broad spectrum control, good on root diseases like summer patch and things like that as well. So that's one newer option when you're thinking about a fungicide program. This is an SDHI called Posterity from Syngenta. That's an excellent dollar spot fungicide. They've got a couple of combo products with with Posterity, the pidiflumethafen, which is the SDHI comboed up with a QOI, which is not going to pick up a lot of dollar spot, but picks up brown patch, things like that, along with propiconazole, that's also going to pick up really good dollar spot control. So that's another good example where you're going to pick up brown patch and dollar spot at the same time. Posterity Forte is a little bit better for Bermuda grass. It has less propiconazole, still going to get dollar spot control. Typically in warm season turf, dollar spot's not a major problem, but in cool season turf, you're going to pick up dollar spot and brown patch again with this fungicide. So that's another option. Acernity, which is a SDHI benzovin diflupyr along with difenoconazole. Again, going to give you dollar spot and brown patch control. Benzovin diflupyr or solatinol, when we first had it in our trials as a numbered compound, it was an absolute world beater on the rhizoctonia diseases. So this is an excellent fungicide for brown patch along with dollar spot as well. And then from FMC, Rayora, another DMI fungicide, which also is going to give you really good dollar spot and brown patch control. The other thing that's kind of nice about the flutriafol that's in Rayora is the backbone of the molecule tends to be flexible. So there's some promising results that even though you might have resistance to some DMI fungicide chemistry, because of the flexibility of this molecule, it may be able to bind into that active site a little bit easier and retain some of that activity, even if you have a little bit of DMI resistance. So that's an excellent thing. And it's also what we would call a safer DMI in terms of its ability to be used along with PGRs like Primo and other PGRs so that you get good summer safety. But you're certainly going to pick up dollar spot and brown patch control with Rayora. And then Kalita is a combo product with fluindapyr, which is an SDHI, along with the flutriafol from Rayora. It's performed extremely well for us in our warm season trials, but we've also seen good brown patch and dollar spot control and things like that in our cool season trials. So that's another option that you have for control there. So thank you again for giving me the opportunity to visit with all of you a little bit.
Thank you, Dr. Horvath, for the time today, and Dylan as well. Appreciate you both coming on to give us a great overview of Dollar Spot. And want to thank everyone for tuning into the FMC First Friday, hosted on the second Friday of this month. And hope everyone has a great weekend.
Thank you, everybody.